Good morning, everyone. Welcome to Tufts. It's great to be here. And so today I'm sharing part of a lecture from a class I recently started for biomedical engineering sophomore majors. And it combines biology and engineering, kind of approaching these topics and how they are applied to research and real life. And so this is an interactive class. So we do use Poll Everywhere quite a bit in my classes for students to be able to put in answers live in the lecture. And so you're welcome to join. You can either go through the website or text my address, and when the questions come up, you can answer them and we can try and solve this murder together. So the crime that we're trying to solve, this is a small village in Wales in England where 90-year-old Mabel Layshorn was murdered. Her murder was brutal but also creepy, and her blood had been collected in a saucepan and showed signs of the occult. And so, we wanted to figure out who the murderer was. There were some tennis shoe marks left. There were blood stains. And so the goal was to try and use the blood from the scene to try and identify who the murderer was. And so we have 4 suspects here. The first is Sarah Layshorn, Mabel's niece, who was her only living relative and who was desperate to inherit from her. We also have Mary Peters, who's her neighbor, and so they had quite a rivalry that everyone knew about in the town. We had Matthew Hardman, who was her gardener, and he came over a lot to take care of her garden. And then we had John White, who grew up in the town and had recently gotten out of jail, and so his parents lived close by. So how do we go about trying to, figure out if any of these 4 people are responsible for Mabel's death? So what makes us unique is our DNA. This is the component that drives everything within our body, and this double helical structure of DNA that makes us unique. But actually, the DNA sequences between you and me is actually quite similar. There's a lot of overlap between 2 individuals and up to about 99.9% similarity between the DNA of 2 individuals. So it's not always that easy just to, figure out the differences between different individuals. And actually, if we look at other organisms, for example, such as mice are really commonly used for experiments in the lab. How similar do you think our human DNA is to that of mice? 92%, 85%, 75%, or 99%? So you can go on the website here, and you should be able to be putting in responses to what you think the answers are. All right. Okay, so unfortunately, having a little technical issue, but that's okay. I can see the results, but they're not posting up here. So currently what we have is a majority around 92% here, and then some down here. So between mice, there's really high similarity. It's somewhere between 95%, in the range of 95%, depending on the studies. Some genes that we have in common with mice are about 99% similar, and some are only about 60%. So there's a range depending on the genes. But there's very high similarity even with other organisms such as mice. So what about DNA is important in terms of studying it? So DNA is composed of these nucleotides, which have 3 components here. We have 3 parts of this nucleotide, and these different nucleotides can be bound together by a covalent bond, which is a very strong bond that keeps these bases together. And each nucleotide is composed of this phosphate group that is negatively charged, a sugar, and also a base. And there are 4 different bases that are available within our DNA, A, T, C, G. And so because of this phosphate group, DNA is negatively charged, and this is an important feature when we're trying to study it. And then DNA is also packed in a double helix structure where you have 2 strands that wind around each other, and you have interactions between these bases. And they only interact in a specific way, where the A base interacts with the T base, and the C base interacts with the G base. And so we always know, if you know what's the sequence that's on one side, you will always know the sequence that's on the other because they're complementary. So within our cells, DNA is packed up into these structures called chromosomes. So DNA, actually, if you spread it out of a cell, can take over a lot of distance. But our cells, we need to pack a lot of that DNA within our cells, and so they're packed in these structures called chromosomes, where you have these molecules called histones, where the DNA is wrapped around really tightly to condense it into this chromosome structure. And so here are the chromosomes that are present within human cells. So each cell, each somatic cell, which is basically all our cells in the body except reproductive cells, have 22 pairs of chromosomes and then 2 sex chromosomes. And so you can see they have different lengths and structures. And again, all cells within our body have all these chromosomes with the sex chromosomes as well. And so these chromosomes make up contain the 25,000 genes that are important for making us function as human beings. And so one main difference when it comes to sex are the sex chromosomes. And so for a male, you would have an X and a Y chromosome, and for a female, you would have 2 X chromosomes. So when we're trying to identify a suspect at a murder scene, we're trying to get DNA, because we know that that's one way we can look at the identity of individuals. And so sources of DNA include any biological material that is coming from the body, because again, all cells within our body have all the same DNA. And so this can be blood, semen, something where you would have cells from the mouth, like a cigarette, or where they were drinking. But of course, these can be tiny trace amounts, right? I mean, sometimes you might have a lot of blood in case of this murder, but sometimes you might just have traces from a water glass. And so a lot of the times, you really don't get a lot of DNA, and so you're wanting to try and make more DNA out of what you get to be able to analyze it. So how do we visualize DNA? So we use the property of DNA that it's negatively charged, and we can put it within a gel, within a solution, and apply current to it with charges on each side, with a positive charge and a negative charge. And because DNA is negatively charged, when you apply a current to the gel that it's on, the DNA will go towards the positive side. It will be attracted to that and move away from the negative side. So we can run these DNA gels where you can include your samples and apply a current to it and see where the DNA goes, how far it goes. We use different dyes to light up the DNA, and so here in these sample lanes, you have one band of DNA here and another band in a different sample. And you can see that they've moved at different places. And that is because they have different sizes. And so because of the gel being a unique composition, if a DNA molecule is bigger, it will have a harder time going through the gel because it's just bigger and it doesn't fit as much into the small holes that make up this gel. Whereas if it's a smaller molecule, it will much more easily flow through the holes, and it will be able to go further down. And so we use a ladder or a set of protein or a set of DNA molecules for which we know the size as a marker for how fast things are moving on our gel. And so you can see here with this ladder, we have molecules that have 1,400 base pairs of DNA. And those would be up here, whereas the smaller ones, like this 300 base pair one, will move much further down. And so you can use this to try and guess and evaluate the size of a particular DNA fragment. So, based off of this gel, what size do you think this fragment is that is in lane 2, based off of the ladder? And so you have 5 options here. Between 300 and 700 base pairs. All right, so I'm sorry it's not showing up. But yes, we have a majority of people saying, 100% saying 580, which is correct, because here your band is slightly further than 600, so it's slightly smaller than 600, so it's 580. That is the answer. So back to the crime scene. So we have our DNA, like I said, but oftentimes there's just not enough DNA to be able to, run it on a gel and see anything, even with the methods that we have. So we need to make more. We need to make more DNA in a controlled way. So how do we do that? Well, the cell has its own process of making DNA. And so this is called DNA replication. So all cells within our body will will multiply to create more cells. And so one cell will copy its entire DNA content and then divide it into 2 equal daughter cells. And this process is what drives human development, where we start, with a fertilized egg, which then, develops into a full human being with thousands and thousands of cells. But this also is important in terms of just general functioning of organs within our body, where we have tissues that need to be replicated, our hair growth, our nail growth, our gut lining, and also in the context of damage when tissues need to be repaired. And so this process of DNA replication involves taking the initial strand of DNA and then separating it, and then making 2 copies of that initial strand. This process is called semi-conservative because we're taking that initial strand, breaking it in 2, and then making a copy of each one, as is shown here. And then we would take these, and separate them, and then make more copies. So the cell has a really good, endogenous way of being able to copy its DNA. And so this is a very tightly regulated process that involves a lot of different enzymes and that regulate this. And so I'll show a quick video on how this process works within a cell. All right, so.
Molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the lead-in strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand.
All right, so this is how this process is done. We have this primer that comes in to tell us where this replication is starting. We have this enzyme DNA polymerase that is coming in and adding the new bases on the other side. And then once you have these different fragments together, you have an enzyme called ligase, which comes in and ligates, as its name would suggest, all these different little pieces of DNA that have been made together to make one long strand. So you have a primer, you have a polymerase that's adding bases, and you have a ligase that brings those pieces together to make all of the DNA that is present in the cell. But so if you didn't have this ligase enzyme in this process, what would happen? Would the strands not be able to separate? Would replication result in a bunch of small fragments? Would the other strand not be made? Would the replication process not even be able to start? So any thoughts on what would happen if you didn't have this ligase enzyme that ligates the pieces together? All right, so what would happen is that you would get a bunch of these very small fragments. You'd get DNA that would be replicated in the cell, but they wouldn't be brought together because the ligase isn't binding them into one big strand. And so this is actually a method, an endogenous process that engineers have taken advantage of to make a method that we can use in the lab to replicate or amplify a piece of DNA in the way that we want. And so this is a method called PCR, or polymerase chain reaction, where we can kind of replicate within a tube with a piece of DNA, a primer that tells us where to start, and a polymerase which makes DNA in a bunch of small pieces in a very controlled way. And so I have just a short video on PCR and how that works.
It's a powerful technique that allows us to make billions of copies of specific pieces of DNA. The first step in PCR is to open up the two strands of DNA to expose the genetic information encoded by the nitrogen bases, A, C, T, and G. In the next step, custom-made DNA molecules help us find the beginning and end of the DNA piece of interest among the billions of letters in the genome. These genetic bookmarks are called primers. The third and final step is to fill in the genetic information between the primers. An enzyme called DNA polymerase adds the building blocks of DNA called nucleotides to create an identical DNA strand. DNA polymerase works in our own cells to replicate our own DNA each time a cell divides. The first step in PCR is known as denaturation and is enabled by high heat. The second step is called annealing, and different primers have different and very specific annealing temperatures, typically between 50 and 60 degrees Celsius. The third step is called extension. DNA polymerase used in PCR works best at 72 degrees Celsius. In these three steps, we've taken our original DNA molecule and obtained an identical copy of a very specific fragment. If we repeat these three steps one more time, we'll go from 2 copies to 4. Repeat it again, and we'll obtain 8 copies. And again, and we'll get 16, and 32, and 64, 1.
All right, so you get the point. This PCR method was developed by engineers taking advantage of an endogenous biological process by engineering the proteins that we make in our cells to put in a test tube with DNA and make a lot of DNA from a tiny piece in a controlled way. And so this method is very commonly used in lots of different contexts, and so we'll see how this can help us solve our murder. So the first we'll look at is trying to identify the sex of the murderer. So as we discussed, biological females have 2 X chromosomes, and biological males have an X and a Y chromosome. And there's a gene called amylogenin, which is involved in tooth development, that has copies both on the X and the Y chromosome. But the version that's on the X chromosome is actually shorter. So a piece has been deleted, and so there's actually a much shorter piece of this same DNA. So on a Y chromosome, this piece is quite long, and then on the X chromosome, it's much shorter. And so, as you remember, we can see different pieces of sizes of DNA within our sample. So we can use PCR to amplify the DNA in this region and see what is present in our sample. Do we have amylogenin from Y and X, or do we have amylogenin just from X? So this is what we did, and so we did a PCR for amylogenin, and we amplified the DNA, and we obtained this gel from the sample. And you can see the data here from the sample of what was obtained. And so we can actually quantify the intensity of the signals, and then put them on a scale to be able to see what size these fragments are. So based off of this, the fact that we got these 2 bands for this gene, do you think that the murderer is biologically male or biologically female? All right, we have some answers coming in. Okay, so we have a majority saying male, and that is correct. Why? Because we have 2 sizes of the amylogenin gene, one longer and one shorter. And if you remember, the longer version is on the Y chromosome, and the shorter version is on the X chromosome. So we have 2 sizes, which means we have one of each chromosome, which means we have XY, which is biologically male. If we had a female, we would only have the one size of the gene. All right, so this allows us to rule out Sarah and Mary, and focus in on Matthew and John. But, you know, that's not enough to solve a murder, right? This is at a small scale. So, you know, how have we, you know, how are we able to scale this up to actually identify differences between people? And so it turns out that there are different regions within our DNA that are very different in between us. And these are called short tandem repeats. So these are sequences of DNA that are present all over our genome that during this process of replication and during development can evolve differently among individuals just based on random evolution. And these small regions have different patterns of length. So for a specific short tandem repeat in one region, you can have, you know, 3 copies, or you can have 4 copies, or you can have 5 copies. So different individuals for a particular region called a short tandem repeat, or an STR, will have a different number of repeats. And so now we've identified about 20 different sites that are these short tandem repeat sites that allow us to distinguish between different individuals. And so if you take the number of repeats at these 20 different sites between me and someone in the crowd, they're going to be very different, and the probability of them being the same is extremely low. And so by analyzing, you know, 10 to 15 to 20 of these STR regions, then we are able to identify with very, very high confidence matching DNA samples, because the probability, again, of my STR repeats matching someone else's STR repeats over 20 different locations is extremely low and likely impossible. So to be able to now solve murders, we do this PCR reaction on these 20 different short tandem repeat regions, and then we compare across different samples. So we did this for 3 of these different STR regions within our suspects. And so this is showing this DNA gel from the PCR on these 3 different regions. And so you can have, if you look at kind of the whole population, you can see that for this particular site, TH01, you know, you can have either 5 repeats, 6 repeats, 7 repeats, 8 repeats, 9 or 10. And again, the more repeats there are, the longer the DNA is, and so it is at a different position in the gel. And so similarly, for this other repeat site, you have about 5 or 6 different options. So what we're trying to do is match the sample, the blood sample that we have, to a specific individual and see if their repeats match. Right, and so this is not, you know, just from a person's repeats, we can't tell who they are, but we can compare 2 samples and see if those samples are the same or are they different. Is this the same person or not? So they did STR analysis for these 3 repeat regions. So we have a positive control, that's just a sample where we know what we're looking for, a negative control where we have just no DNA. Then we have our evidence sample. So this is the sample of the, you know, who we think is the murderer. And then we have the victim reference sample, because the issue with some crime scenes is sometimes, obviously, there's victim sample blood present. So we need to make sure that we're comparing to that as well, to make sure we're not just identifying the blood of the victim. And then we have our two potential suspects. We have Matthew Hardman and John White. So what we want to do here is compare the number of repeats, these green peaks here and here, between these two and the evidence sample, and see which one matches. So based off of this, I'll leave this for a couple of minutes. Who do we think is the murderer? Is it Matthew Hardman or is it John White? Again, we're comparing the peaks between this sample, and then which one is closest or is identical to the evidence sample. So take a look, and then you can vote on the next slide. Who do you think did it? All right, we have some votes in, and we have Matthew Hardman being the murderer. That is correct. So if you look back at these gels, right, you can see that here we have these two peaks that are kind of further apart, and here again, this sample has two peaks that are the same, identical as here, versus the peaks for this sample are shifted. So this allowed us to kind of rule out John White as a suspect, even just from the first sight. But of course, the other ones also match as well. All right, and so Matthew Hardman was eventually arrested. The probability, again, of based on doing 20 of these STRs of him matching with someone else is very low. And so that's why when we're trying to solve crimes, there's a database, right, of DNA evidence from particular individuals, and police are always trying to analyze the DNA of a crime scene and see if it matches with something else, right? It's all about matching. We can't just identify someone based off their DNA, but we can match them to a known sample. So this can be used on a much larger scale in terms of sequencing. So all the changes in our DNA lead to changes in protein that affect who we are as individuals. And these changes in our DNA are called SNPs, or single nucleotide polymorphisms. And this is actually what is studied in services like 23andMe or Ancestry, which now have millions of people signed up for them. You can also do it for your cats and your dogs to identify their lineage. And you can get a lot of information from this within the different, that they provide. Some are health-based, some are more characteristics and associations. But basically what these sites are doing a bunch of PCRs on a bunch of places in the DNA where we know a difference in a sequence can impact a certain output. So for example, for eye color, there's actually a specific DNA sequence that reflects your eye color. And so for I did 23andMe, I have 2 G base pairs at this particular site, which means that I'm gonna have blue eyes. So, but someone who has brown eyes will have different bases shown here within their DNA sequence. Now, what really becomes interesting is that now these, when you provide your samples of DNA, you can actually also find relatives, right? So there's a lot on Ancestry in terms of where you come from. So this is my heritage. But it also will look up a bunch of my relatives that are related to me genetically, because there's a lot of these features like our eye color, our height, things like that, we inherit from our parents. And so these are traits that can be found within our relatives, and so they can connect you with relatives, of different closeness within their data set based off of their samples that they have. And so this now is actually used to solve murders. You may have heard of the Golden State Killer, which killed lots and lots of people in California in the 80s, and they could never find this person. They had DNA frozen in a fridge, but they could never match it to the right person. And so what the police did is they created a profile on, not one of the bigger ones, but this was a while ago, on this one called GEDmatch, which is, similarly ancestry repository. So they created a profile, they sent in the DNA from this DNA they had isolated from a crime scene that they were pretty sure belonged to this one guy, but they couldn't prove it, and they created a profile for him. And then that website connected them with relatives of this person. And then based off of that, they were able to, piece together with relatives and information, who this person likely was. And then they stood outside of his house and they stole his trash and they got DNA from that and they matched it to the original DNA from the collected crime scene. And so this is kind of a new way that we are solving murders by taking advantage of this DNA that's deposited to create connections and find more information about people. And this is not just being used for murders, but also in the case of adoption cases and things like that. All right, so to summarize, we've talked about how DNA can be isolated from crime scenes, although a lot of the time there's not enough. It needs to be amplified, and engineers have developed a method called PCR, which allows us to generate a lot of DNA from a sample in a very controlled way. We can run it on a gel and separate it out and analyze that data. And we can do this on lots of different sites, either these short tandem repeats that allow us to identify individuals, or on features that we know are associated with particular traits to learn more about different individuals and also find relationships and identify individuals for identification. So with that, I have a few minutes for questions, and thank you.
Hello there.
Hi.
Question, how long in real time does this actually take from the time you get a sample to the time all these methods are completed? Because it seems like in the news, it takes like months or years, but how quick is it?
I mean, it depends, for specifically doing like the SNP work or like doing the sequencing of those different sites, like it doesn't take very long. The experiment in itself will, take a day probably. There's definitely a lot of data analysis behind it, but, it's definitely not that long of a process. We are now getting really good at doing this really well because these methods have been around for a while. But, there's a lot of analysis and kind of data input and then on that end as well. But, generally we're getting much, much better at it. Like now we can replicate the whole genome or whole DNA within just a couple hours for a couple $100. So the technologies have improved so much in that area that, we're getting pretty quick at it.
Hi, I have a question, just based on things I've seen on sort of TV shows. So if it was found out that Matthew had an identical twin,
Yeah.
would he be like they couldn't figure out who did it, or is there a way to determine if it was truly Matthew? Yeah,
so you can then look at, a few more features and places within the genome as well, and also compare to, it often can be helpful in terms of looking at the parents as well to see that information and because we know all of our DNA is inherited from our parents. So, and then there might be other factors that come into play relating to the case itself and more kind of physical evidence. So yeah.
Hi, thank you. I was curious, you said you found DNA on the knife from the criminal. Is that possibly skin cells, or must he have been bleeding? And do you find the same DNA in skin cells as blood?
Yeah. So it could be definitely lots of things, but yeah, we have all the DNA is the same in all the cells in our body. So all the DNA will be there. These cells are different because what's made from the DNA is made in different ways into different proteins, and that what gives function of our different cells, even though they have the same DNA. But all cells from an individual the DNA is the same, except for the reproductive cells.
Hi. If the lab wants to cheat, can they contaminate it in a way where they can stick like we have a different color than the real one?
Yeah, I mean, I am not a forensics expert or anything, but yes, obviously this contamination can happen and that can be a big problem, even in terms of different steps along the way, at the crime scene, things like that, for sure, that can happen. DNA is very stable and it lives around in lots of different places, and so it's quite easy, or it can be quite common for DNA to contaminate things even within a lab. So that's why, generally, we do run a lot of controls, a lot of reactions to make sure that we're doing things in the best way. But yes, I'm sure things like that can happen. Yeah.
Yes, my question is, how is the physical replication actually done in the laboratory? You talked about replicating and enhancing patterns.
Yeah, so we basically just take the DNA from, that we can isolate from a cell, for example, and we then buy an enzyme, this DNA polymerase, that, we can buy commercially that gets activated at certain temperatures. And we also buy a piece of DNA for the specific sequence that we want to amplify. We put all that together in a machine, and we have, as you saw, there's a temperature control that allows us to, we want to use temperature to kind of separate the DNA and then activate different parts of it. So we can set it in a machine where you say, I want you to go at this temperature for, 5 minutes, this temperature for 10 minutes, and then come back down, and then it repeats over and over. So we have great machines where you set up all the temperature changes you want, you mix everything together in a solution, and then, an hour and a half later, you could get all your DNA that you've amplified from that particular sequence.
Oh, I have one more question. Someone earlier brought up the sequencing of the DNA of twins, and so I know that the environment of an individual plays a role in the degree to which certain genes are expressed based on how tightly it's wrapped around the histones. So I guess my question is, in that situation, is there a way to measure how tightly bound the DNA is around the protein histones, or is there just the sequencing to look at?
So there's other methods in place. So here we're focusing really just, we talked about methods for DNA, but we have lots of other sequencing methods and analysis methods to study the epigenetics, which is what you're talking about, and the regulation of the DNA in that way. So you'd just be looking at completely different molecules. But yes, it's definitely doable, absolutely, just not with this method.
And we have one more here. This will be the last question so we can get to the next sessions.
Obviously, this technology opens up a lot of ethical questions, right?
Yeah.
Because the same technologies can not only be used to identify murders, it could be identified to look at disease genes in individuals.
Yeah.
So what prevents, for example, an insurance company to submit a sample of somebody to figure out whether or not, for example, that person is at risk for cancer? So
it's a huge problem, absolutely, that's being discussed. And a lot of the times too, so for certain gene mutations like BRCA, which is a common one for breast cancer, there is something we can do about it in terms of, monitoring patients closely. But for some mutations, and actually on 23andMe, they give you a couple genes that are associated with Alzheimer's, and they can give you increased risk. And so, but at this point, there's really nothing actually we could do. If someone had that risk and exhibited no symptoms and went to their neurologist and was like, I'm predisposed to this, what can you do? And there's actually nothing they can do right now. So, it doesn't necessarily give that extra information. And so, it's a huge, huge problem in terms, sometimes knowing the sequence of the DNA is very helpful in identifying treatments for certain diseases, and knowing that right away, can really help a lot. And then sometimes it just opens more questions, and we can't always predict disease progression too, so some of these markers, may not actually lead to anything. So it's very complicated. Yeah, it's it depends for what diseases, what context, kind of what information, and what we can do clinically, and so there's a lot of factors involved in that.